Hello viewers, today I am going to present before you the replication in viruses. I am Dr. Seba, HOD and Associate Professor, Department of Zoology, Gurucharan College, Silchar, Assam, India. Before going to start the topic, let me talk about something regarding this presentation. Actually, it is a part of the initiative taken up by the Zoological Society of Assam for undergraduate students who are pursuing zoology honors in different colleges of different universities of India. Learning outcome of this presentation are Introduction to Replication in Viruses, Types of Replication Cycles of the Viruses, Key steps in viral replication cycles, mechanism of replication of viruses inside a host cell, and comparison of lytic and lysogenic cycles. Now, let us talk about the viruses. All of us know that viruses are small, submicroscopic intracellular parasites which contain either DNA or RNA genome surrounded by a protective virus coded protein coat. A complete virus particle is called a virion, and the main function of a virion is to deliver its DNA or RNA genome into the host cell so that the genome can be expressed by the host cell. Now, the replication of virus is a complicated process. Viruses never reproduce by division. They are replicated by a process in which all components of virus are produced separately and are assembled into intact virions. For replication of virus, host is necessary, and viruses are host specific. The host may be a bacteria, a plant, or an animal. The two cycles in the replication of viruses are the Lytic cycle and the lysogenic cycle. The Lytic cycle involves the reproduction of viruses using a host cell to manufacture more viruses. The viruses then burst out of the cell. Secondly, the lysogenic cycle involves the incorporation of the viral genome into the host cell genome, infecting it from within. To understand life cycle of a virus, we'll have to know about the structure of a bacteriophage. Let me talk about a bacteriophage. Viruses that parasitize the bacterial cells are called bacteriophages or phages. The phage meaning to eat. The phages have specific hosts and they are of variable shapes, sizes, and structures. The most widely studied phages are T-even bacteriophages such as T2, T4, T6, etc., which infect the colon bacillus. T4 bacteriophage is a large-sized tadpole-shaped complex virus. Its capsid comprises of a head, a short neck with collar bearing whiskers and a long helical tail. The tail is made up of thick and hollow midpiece, a hexagonal base plate or end plate to which are attached 6 spikes and 6 long tail fibers. The midpiece consists of a central hollow core and a spring-like contractile sheath which comprises 24 rings of hexameres and remains helically arranged around the core. The T4 genome or chromosome is a single DNA molecule which is 60 micrometer long, linear, double stranded, and tightly packed within the head of the fudge. The key steps in the viral replication cycle are attachment, number 1, then number 2 is penetration or entry, number 3 is uncoating, number 4 genome replication, number 5 assembly, and number 6 is release. The Lytic cycle of replication in viruses is a 6-stage cycle. The 6 stages are number 1 attachment, number 2 penetration, number 3 uncoating, number 4 replication, number 5 assembly, and number 6 lysis. The first stage in the replication of viral cycle is the attachment. Firstly, a virus is host specific and it enters into the host cell or target cell which is specific to it. Secondly, this event is electrostatic, which does not require any cellular or metabolic energy, and virus exhibits cellular tropism. That means the ability of different viral strains to infect different cell types or tissues. Virus has host range which may be narrow or broad. For example, HIV is an example of broad range virus. A virus uses receptors and antireceptors for attachment and entry into host cell. Cellular receptors and antireceptors are mostly proteins, but sometimes they may be glycoprotein, carbohydrates, or lipids. The presence of virus specific receptors is necessary. For example, HIV has CD4 receptor, rabies has acetylcholine phospholipid. The second stage in replication of viruses is penetration or entry. Penetration is energy dependent process. Virus may penetrate into host by endocytosis. That means it is the process of actively transporting molecules into the cell by engulfing it with its membrane. Secondly, it can also penetrate by the process of translocation, which is the movement of a molecule across a barrier or between cytosol and membrane surface. Thirdly, virus can also penetrate by the process of fusion. Fusion is the process or result of joining two or more things together to form a single entity. The third step in viral replication is uncoating. It refers to the removal of capsid, thereby releasing the genome into host cell. The virus genome is transported to the site where transcription or replication can begin. In some, there is no degradation of capsid as capsid proteins play a role in viral transcription and replication. The fourth step is genome replication. Viral genetic material or genome is multiplied within the host. Simultaneously, viral structural proteins like capsids are also synthesized. Type of genetic material varies from virus to virus. Fifth stage in the replication of virus is assembly. It involves the collection of all components necessary for formation of a virion. It takes place at a particular site in the cell. For example, in pox viruses, assembly occurs in the cytoplasm. In adenovirus, it occurs in the nucleus. The sixth step in viral replication is the release. Newly formed viruses are released to outside of the cell either by lysis as in bacteriophage or by budding as in retroviruses. Generally, non-enveloped viruses release by cell lysis, that is breakdown of the cell membrane, which results in the death of host cell. Release of virus by budding may or may not kill the cell. In this picture, you can see that a virus is released either by lysis or by budding. In the lysogenic cycle, the phage DNA first integrates into the bacterial chromosome to produce the prophage. The daughter cells can continue to replicate with the prophage present, or the prophage can exit the bacterial chromosome to initiate the Lytic cycle. Let us compare the two cycles, that is Lytic cycle and lysogenic cycle. The Lytic cycle involves attachment, penetration, uncoating, replication, assembly, and release of the virus. On the other hand, in lysogenic cycle, a virus can replicate its DNA using a host cell. Now we have arrived at the end part of this presentation, that is conclusion. It may be concluded that there are two cycles in viral replication, such as Lytic cycle and lysogenic cycle. Lytic cycle involves 6 steps: attachment, penetration, uncoating, replication or biosynthesis, assembly, and then release of the virus. The lysogenic cycle is a method by which a virus can replicate its DNA using a host cell. These are the references or bibliography I have consulted. Thank you so much for tuning into our presentation. Your valuable feedback is welcome. You can email your feedback to my email ID rashaba1@gmail.com. You can subscribe the channel if you like. Thank you. Thank you once again.